All right. Yeah, I want to thank the organizer. This has been really a fun meeting. And also it's like really nice to, I guess why it's one of the nice, probably one of the few perks of getting older is that you get to meet all the wonderful people and then you meet them again and it's nice to share. There isn't much else that is good about getting old. So at least I'm enjoying that part. All right. So today I'd like to talk about what is going to be obviously an interesting problem, which is the fact that during appendage regeneration in zebrafish, things don't only grow back, but grow back to the same size and shape that they had prior to amputation. So generally, this is the same problem that many in this room are interested in, which is how do organ and tissue grow to the correct size and shape, and this is referred as morphogenesis. But what is interesting is that some organisms, but not all, have also a great ability to regrow this tissue back once there is a severe injury. Our organs are also able to rejuvenate and get regenerated. But really, things like a planaria, right? You can cut it in half and you get two planaria, able of doing even whole body regeneration. And the fish can regenerate spinal cord, heart, lost appendages like fins. Well, if I cut my finger, I'm not going to get a new finger back. So this is sort of both a very interesting biological problem and one that people are excited about because maybe if we understand why the fish does it and we don't, we could eventually one day use that knowledge. The question we are interested in are the same one that everybody in this room is probably interested in. Can we learn something about the molecular mechanism that encode tissue size and shape? Can we describe the cellular dynamics that drive regeneration? And we have also been interested in this question of how can events that essentially happen in individual cells on very fast time scales be integrated over the millimeters and weeks, because this regenerating system can be big and regeneration takes a long time, right? I work also on early fly embryo, and there we make 1 to 2 hours movie, and we got all the data we want. This is a month process, so there is an issue of bridging time scale also. So the first study I told you about, you probably didn't know coming in today that you'll hear not one talk, but you talk about scales. But it's zebrafish scale regeneration. This was the work of a wonderful postdoc, Alessandro, now is his lab at University of Geneva. But it's all published work, so I'll try to go through it quickly, but I'm happy to talk to anybody during the breaks if you're interested in this. In fish, the scale is actually a piece of bone. It's a flat bone disc, so a lot more similar to our facial bones. And it's mainly secreted by this continuous layer of osteoblasts that sit underneath the bone. There is some osteoblasts also on top and more sparse osteoclast. And it's the osteoblasts that secrete the bone matrix and the osteoclast that would chew it up. And so you get the proper matrix. And the scale is inserted in the dermis, is in touch with all this basal epidermis and other cell of the dermis, and it's covered by epidermis. Experimentally, the way we do the experiment, or Alessandro and others do experiment, is that they go in with a forceps and pluck a bunch of scale and they watch them grow back. And so this is what it will look like if you have an osteoblast-specific promoter driving GFP. You can see all the scales in this beautifully hexagonal array, and then you plug them, they're all gone, and within a matter of days, they get all the scale back. And what happens on the cellular level is that, at best as we can tell from many experiments, once we remove the scales, we really remove all the osteoblast. And so there is an initial differentiation phase in which cells, and we don't really know the origin of those cells, but some cells probably from the dermis transdifferentiate and give rise to an initial pool of osteoblast. That lasts about the first 36 hours of the process. Then from day 1.5 to day 4, there's a lot of proliferation of this osteoblast that makes, if you want, a scale primordium. And then the bulk of the growth of the scale is actually hypertrophy. These are cells just getting bigger without replicating their DNA or without dividing, but getting really, really big, and that's what drives the growth of the scale. And this is the phase that Alessandro worked on, was what controls this hypertrophy and this growth. And how is that organized in space and time? we have reason to believe, maybe not surprising from the work of many prior to us, that FGF signaling is very important for tissue growth in regeneration. And here I'm just depicting the usual cascade. And our approach, inspired by some work we have done in embryo, is that when you want to look at signaling, if you can, you want to sort of use some of those reporters that will allow you to look at the signaling activities in a quantitative way in vivo. So these usually involve either a threat sensor or, in this case, this translocation reporter, in which there is a docking site for ERK in this engineered protein that was made by Sergi Rego when he was a postdoc with Marcus Covert. And what this docking site essentially gives you specificity toward ERK. And then if you engineer NLS and NES sites, you can do something so that when this peptide is phosphorylated, it likes to sit outside of the nucleus. And when it's not phosphorylated, it will sit inside the nucleus. And if you then fuse it to a fluorescent protein and can measure how much of the protein is in the nucleus and outside of the nucleus, you get a proxy of kinase activity. So Alessandro could take images like this. So when you see a lot of protein in the nucleus, you can say there is very low ERK activity, and you can do some image analysis trick to turn this into a quantitative map. And when the nuclei appear as empty or ghosts, and when all the protein is on the outside, the ratio of cytoplasmic to nuclear protein will be high, and then it's indicative of high activity. Then what we do is that, this process of regeneration takes weeks, and you can keep a fish anesthetized for weeks. It's going to die. So what Alessandro and what we normally do in the lab now is that you will anesthetize an animal, find a particular scale, you image it, and then you go back, wake the animal up, let it swim around, and then go back 6 to 12 hours or the next day. And anesthetize again, find the same scale and image it that way. So these are movies, but where a time frame is every 6 hours and the fish is swimming in the meantime. But then, and I don't have the time to go through all the steps that go into this, but after some elaborate image analysis, Alessandro was able to really generate those heat maps of activity where every single cell or nucleus in the scale is now color-coded for its ERK activity. And what you see is that there is these concentric rings of activity. But what was particularly exciting is that when he looked at this dynamically, he discovered that those rings are not statics, but there's these concentric waves that propagate through the tissue. Yeah.
Like the place that's source of that?
Yes, that's a beautiful and important question. So there is the waves always seems to start at the same place. We do not know what are the ligands that drive it or the cells that drive it. But one experiment Alessandro was able to do is remove a scale, turn it around, because it starts a little off center. So if you remove the scale, turn it around and reinsert it in the pocket, then it starts where the scales touch the dermal pocket and not where it started before you remove it. So we think there's probably some signal that comes from the dermis underneath that initiate where the wave starts. But we don't really know what the ligands are or the cells that do the signaling really are yet. At best, as we could tell, there is no difference between those osteoblasts and the remaining osteoblasts in the scale. Yeah. The way that we think that those waves are generated is through probably the simplest model, and we have some evidence that those have property of reaction diffusion, excitable waves. And the way that you could get that is if there is a positive feedback, so that when ERK is active, it triggers either the release of a ligand or the activation of signaling that can stimulate the next cell. And then there are also these inhibitors that Alessandro now from his own lab has a preprint where he has visualized it that come on with some delay relative to ERK activity and shut it off. So this is very similar to an action potential travel down axon, where you can stimulate the next region, but there is a refractory period behind because it's loaded with inhibitor. So this, yeah.
And can you control that refractory period?
Yeah, it's doing genetic manipulation and things a little tricky in this system because it's all adult and genetics is complicated. We might have an optogenetics line to control ERK that probably works, so you could start doing that. There may be ways to, now that we know some of the inhibitors that are expressed, there may be ways to manipulate that expression, but we haven't done much yet.
And is that excitability present even at the local single cell level? And then there is a spatial aspect to this, which you also need because you need enough of a nuclear size, coming back to the question of.
Right. So we have not tested yet. That's one of the reasons why we made this optogenetic line. And so ideally, yes, what we would like to do is go and activate region of different size, because as curvature, as you know, right, of course, inhibits the propagation. In our model, we have a mathematical model of this that suggests that you need an activation of about 50 micron to be able to propagate a wave out, which would be a nice hypothesis to test, but we haven't tested yet. But I think optogenetic would be the right way to do it.
And this precedes hypertrophy.
So what we found, essentially, every time a cell sees ERK activity, it grows. So there are essentially, if you just do some cell tracking and then compute a vorticity field, what you see is that the expansion kind of slightly follows where the ERK activity is. So we think that a cell that sees ERK is growing and the other cells aren't growing. And this also leads a little bit to this idea that the reason why you want to use waves to grow, we could do some other experiment where we forced the old scales to have ERK activity in synchrony, and then they grow less. And we think it's because of some mechanical feedback, in which when they're all trying to grow, that inhibits their ability to expand. But if you're only growing in a region at the time, it's easier to propagate that stress.
And cytoplasm to water volume ratio changes enormously during this hypertrophy?
We don't know, right? I mean, the hypertrophy really triggers a big growth of the cells, but it takes forever. So for every wave, a cell only grows 15 to 20%, but you have 6 of those. And then the cells get 3-4 times as big. But so it's very, very hard to really know, because you really have to look at cells from one day to a week later. From one day to the next day, they're going to look about the same.
Is there any relation to the kinds of processes associated with I forget what, the plates associated with limb growth during adolescence, which also happens through hypertrophy, and it's mostly associated with volumetric changes without cytoplasmic changes? So smaller driven, is that?
Yeah, we haven't looked. My guess will be that this growth is accompanied by the fact that they're making more protein, and so they're actually growing in dry mass, but we haven't tested. It's just a guess at this point. ERK is known to trigger translation and biosynthetic capacity, so that seems a reasonable hypothesis, but we don't know. All right, so in this process, there's about 6 waves, and the beginning they come once about 1 a day, and then it slows down, and there is a very good tracking of the overall area of the scale with this number of waves. And then the other experiment that also suggests that really the function is to trigger growth is that what Alessandro could do, he could go in with an FGF inhibitor just transiently, just for a few hours. And the wave stops, and for a reason that we don't fully understand, it takes about 3 to 4 days until a new wave shows up. And in these 4 days, the scale kind of keeps its size, and then as soon as you get a new wave, then you start growing again. So there's a bit more in this paper that you can read, that was published about 5 years ago. We were excited because, as far as we know, is one of the first example, if not the first example, where signaling waves were clearly documented as a mechanism that controls regeneration. And of course, you can speculate that is a mechanism to spread information much faster than you could do by diffusion in this large tissue. Yeah.
The entire thing to like a disc or something, so that it can't go outwards, but it can still do the cell division. What happens?
Yeah, that is hard to do, right? So this, I always show you a single scale, but that's because it's computationally extractive. We are doing this in the fish, right? So as the fish is swimming around, so probably one could try to clamp a ring around it and having not fall off, but I don't think that the fish is going to be up here. It's going to be easy to do. So that mechanical constraint is not easy. As far as I know, nobody has been able to grow those scales in a dish and get, but no, I also, as far as I know, nobody seriously tried, right? I mean, with this organ, I feel now it seems that if you put the effort, many things can be grown in a dish, but we haven't tried. So this is not where we put that effort. So So yeah, all those experiments would be possible if we could get an in vitro system. What would I mean, I would just expect that the buckle in the other, the buckle in the other dimension, yeah, but I don't know if that will lead to something interesting or not. Yeah.
Does the boundary curvature or shape affect the shape of?
Yeah, you would expect that it could, but it's a little tricky to measure it very, very accurately what happens as they get to the boundary. And these are small layer of osteoblast at the boundary are also believed to be a little different. So we haven't really looked. There is an influence of curvature on how the wave propagate, and that we could test because these waves are also present in juvenile fish, which are much, much smaller and have very small scales. So on those small scales, if you look at the speed of the wave, it's very, very slow. And the reason is this because the wavefront is curved. So there is some influence of the curvature on how the wave propagates, but we haven't applied that to think about the boundary. All right, if there's no more question, I'll switch now. So these really got us to think a lot about how tissue grow during regeneration. But the question that we have now decided to focus on, which I think is more interesting and it will obviously be obvious from these, is the one that they don't only have to grow, but they have to grow to the right size and shape. And this is what I like to call geometric memory, or in the field is also known as positional memory or position information. But what you're looking at is a fish, and if we zoom on the caudal fin, if I go in and amputate it, you could see that within about 3 weeks, you get a new fin that has regenerated. But what is exciting is that if I were to trace the pre-existing shape and the regenerated shape, they are very similar. So somehow there is some information that tells the fish where you amputate it so that they grow back essentially the right amount. Yeah.
There are two fish tails, two different fish tails. Would this look like the regrown? Would the variability between fish be comparable to the variability in the regrowth that you're observing or not?
Yeah, it's a little tricky because these are adults, so comparing a fish to the other depends on how they grew, how much they ate, and the density at which you grow. So that comparison isn't easy. What I could tell you is if you try to put a number with the precision with which they regenerate, is 5 to 10%. So it's not perfect, but it's pretty good. So I think the experiment you're doing, one will need to really be sure that you grow them in very, very controlled conditions to really be able to compare. And I don't think anybody's done. Yeah, I go there and then I'll come to you, Olivier.
Is P of the regrowth age dependent? So the older they get, the less accurate it becomes?
If they are above, so fish are considered adults at 3 months, and usually we are supposed not to keep them after 18 months. Most people in the lab, I think, do. And then I get nasty emails. But I think the ability to regenerate get worse, but I think it's probably not a linear decrease, pretty switch-like. So essentially, at some point, they become old enough that they're bad. But until, if you take a 3 months or a 12 months fish, they're going to regenerate about the same efficiency. Yeah. So just not too old, and they're finally.
I did never realize, but there's an interesting bilateral symmetry of the fin also. Look at the pigments, for instance.
Yeah, that is true, right? It does look like those patterns are symmetric on the pigments. And I don't know if it's known where does that come about. The pigments do not perfectly regenerate, I don't think. So if we look at the pattern they get back, it's not quite the same one that they started with. So I do not know. In terms of why they regrow more or less symmetrically, if I get to the end, what we are trying to say, probably if there is one thing that I feel is probably exciting about this talk is that we think that somehow they read the geometry of the uninjured structure. So as long as you have laid out a bilaterally symmetric structure, you're going to get one back. But let's see if we can get there. Was there another question? Yeah.
You tell us about this. If you cut at different locations?
Yeah, it's in a nutshell, all that they care about, and I'll show you some really nice data collapse. You can collapse all the data just by knowing how much you removed. There is no doubt about that. And that's one thing I feel pretty confident because we have looked at hundreds of fins. And if I tell you length amputated, you can collapse all your data. All right, so what is the anatomy now of this fin? So these are long bones, and the bones are these two open hemicylinders, which are depicted here in red. So they're osteoblasts. Then a very important player are these mesenchyme or fibroblasts, which early in regeneration near the tip is also known as the blastema. In the fish, it's a little more lineage restricted than it is in things like axolotl, where it's really believed to be a lot more pluripotent and drive different cell types. The fish fin is a lot more restricted, meaning that most osteoblasts come from less mature osteoblasts. The mesenchyme come from mesenchyme, and epidermis comes from epidermis, but it's also covered from epidermis. I'll start with osteoblasts because we had the lines, but if I can make it, we'll talk about the fibroblasts, which is the newer stuff. So I really hope to get there because on this one, there's a pre-plintule also. But then again, you amputate, it grows back. One easy question to imagine is like, how do the osteoblasts that sit on a lateral position know that they need to grow more than the one that grow in a more medial position? This is something that was already sort of known. We've done some quantitative experiment. I'm not going to show. There is not much contribution from cell rearrangement, cell moving around. And early contribution, maybe a differentiation, making those domains correctly, and we'll talk briefly about that. But the bulk of what drives the tissue growth is proliferation. So we wanted to be quantitative and characterize proliferation. And again, in this system, it will also turn out that FGF and ERK are important. So what we did now is that instead of just imaging this sensor, we are also going to image a cell cycle reporter. And the way that this works is that people took a piece of a protein called geminin, which is involved in DNA replication, and that piece is targeted for degradation by the anaphase promoting complex, which is active in G1. So in G1, the protein is degraded, but it's stable in SG2M. So if you drive it with an osteoblast promoter, every osteoblast that is positive for this protein, it means that it's proliferating. And you get images like this, where you have these nice fin rays that are growing, and you can see both ERK activity and quantify proliferation.
What does color indicate?
Here, color indicate the activity of that kinase ERK. So yellow means a lot of activity, and blue means lower activity.
So it's the tip which is mostly active.
It's mostly active at the tip, and we'll talk about
that characteristic scale.
Yeah, that's probably most of what I'll talk about in the next 15 minutes. But the first question we asked was, is there a difference in how lateral and medial ray proliferate? This is just a movie where Ashley, the postdoc who worked on this, every 12 hours image how these rays grow. And then what she could do is to do some image analysis. And here she divided this into 3 groups where she cuts less, medium amount, and more, amputates more. And you can clearly see that the more tissue you remove, the more proliferation you have. But what is very interesting that gets to this question of what do they really care about is that you can perfectly rescale all this data into a single, more or less, relationship. If instead of plotting the number of cells that are proliferating, you plot what is the fraction of cells that are proliferating. So how many cells are in the regenerate, how many cells are proliferating, divide by how many cells are in the regenerate. And instead of time, actually, it turns out the fraction regenerated. So how much you have grown divided by how much you removed is the best predictor. And then everything falls into this nice relationship. So there's some scaling here. There's something that is setting that proliferation domain that's somehow reflective of how much tissue you remove. How can this be controlled, right? What controls which osteoblasts proliferate? Not surprisingly, it's again FGF and ERK activity. If you inhibit ERK, you don't add any bone, while normally in a control DMSO treated, you add, in, at least for this fish in this condition, between 150 and 200 micron of bone each day. Yeah.
When you did the amputation, you amputated the entire fin in one
Yeah.
shot. If you amputate different at different times, different parts at different times, is there a kind of heterochromy and a catch-up in that they will all end simultaneously but grow at different rates?
So, yeah, so we haven't done the experiment of cutting at different time. You can cut different amount.
That I understand.
Yeah. So most of what we know, so I don't know that anybody's done the experiment you suggest. There's no reason why we cannot do it. The view in the field is that every ray is independent, and that comes from, these are very difficult experiments to do, but some people did manage to do transplantation experiment. So if you take a lateral ray, you move it to the middle and you amputate it, then that ray grows, but it doesn't influence its neighbors. So there's very, very little crosstalk among them.
Understand what you just said, because if you cut it vertically, the long rays have to grow longer, the short rays have to grow shorter. So each one of them is different. You're saying that they all grow at different rates.
No, they grow at different rates and they finish at around the same time.
Okay, that's what, yeah. So,
yes.
But now that's because you started at the same time.
That is a
My question is, what happens if you're not?
Yeah, I think we should do that experiment. We haven't done it. So yeah, my guess will be that because there is little evidence that they influence each other, once you cut them all together, they seem to be little influence from each other. One possible assumption is that the same will happen if you cut at different time. Everybody will go as, but you are right. I mean, and it's an easy experiment to do, so we could test, and I can let you know in a couple of months what we find. Yeah, no, it's actually a good question. Maybe we'll find something that we did not know. All right, so again, we have the ERK sensor, so we can look. And if you look over time in this system, there's no evidence that there is waves. And actually, the patterns are a lot more random. And that is because if you focus on an individual cells, there are actually there's some oscillations that we are going to now average out and try to curate. So we'll work mainly on cross-grain pattern. And when you do that, the biggest pattern that emerges is that there is actually gradients of ERK activity. So the activity, it tends to be higher near the tip, as Ma also noticed, and lower near the amputation plane, and there is those gradients, but they are very dynamic. And so we felt like we need a way to describe, a mathematical way to describe those gradients. Do you have a question? This is an interesting, we don't really know. We think it's probably mechanics. So as the ray grow and expand, they tend to bifurcate. As they regenerate, they tend to bifurcate later. The simplest possible mechanical interpretation could imagine is that you are extending the surface. And if you want, these bonds are really structurally keeping the thing together. And at some point, bifurcating them probably helps with that structural ability. It's known to involve sonic hedgehog, but really what is the trigger and what determines exactly when and where they happen is not known.
There, they should then care about the labels, right? If you have to fill the intermediate gaps between the rays, there's too much empty space in the middle, then the high density of
so if that bifurcate, yeah, I mean, I would imagine that if it's a mechanical signal, that it may be the same sensed across all the ray because it's really a big geometric property. But yes, this is a question that comes up all the time, and we have some data on it. It's just no student or postdoc made it a priority yet to work that out. But it's a very important question. I agree. All right. So what we okay. So I forgot about this. Right. This is another slide that helps to make this point that ERK is really what drives proliferation, because we can correlate on average in given region how much ERK activity is there and how much proliferation is there. It's noisy, but you get two things from this. One is that there's clearly a relationship. The probability of proliferation is higher the more ERK activity you have. But also this relationship is pretty much independent on how much tissue you remove. So as you can see here, we have about 6-fold difference in tissue that you remove. But more or less, a cell only cares how much ERK is in that cell. It doesn't care if it was you remove a millimeter or 5. It's going to have the same probability. Another way to say that, it suggests that there must be more ERK activity the more you cut, but how that comes about. So the goal then became, we thought that what we really needed was a mathematical formalism that told that how much ERK is at every position, at every time point, and that could also though be a function of how much you have grown out, what I call L here, and how much tissue you remove, that is called L bar or L amputated before. But what we realized is that while this could be a very complex function, Well, if we just talk about this fraction regenerated and position in normalized coordinate, then what you get is that you can actually express this entire dynamics as the product of two functions. One function that just cares about fraction regenerated, which if you want encoded time, and then a function f of u, which actually is the same for every ray at every time point that encode those gradients. The way that I'm going to try to convince you that this is true is to reverse it. We, of course, looked at the data and guessed it. But if I assume that this is true, I can then derive some conclusion from this. The first one is that if I integrate this f of u out, because I say this is the same at every time point for every ray, that's just a number, which means that average ERK activity should only depend on fraction regenerated. And this is what you get. So it's a pretty decent collapse. So it depends on how much tissue you remove, all the data seem to collapse on this linear relationship.
And those two outliers at 0.1, 0.2, what was happening there?
Yeah, so there is so it takes about 3 days to make for this cell to start expressing this austerics promoter. So those I think were caught. They were on their way to become fluorescent, and they were got there. So it was, and there is the lateral rays when you cut a lot take a little longer to get there. So there is, I think it's just a technical detail. So how do you get that f of u? Well, then if I take the ratio of this, so if I take my ERK activity and divide it by its average activity, the A of phi should cancel out, and I should only bring the spatial dependency. And again, by the way that these things go, this is a decent collapse. And so what this means when you put this back in normal coordinate is that this is what ERK activity does. It starts off being very high and steep, and then over time, the gradient both flattens out and extends. And so essentially, it almost acts as a ruler that is telling you how far you are. And once you eventually flatten out enough, you get below threshold and nothing grows. What also that means, because it was all done in normalized coordinate, is that the same behavior will be true independently of how much tissue you remove. So this will be the behavior in one ray. But if you look at a ray that was much shorter, you will have the exact same gradient, just compressed, it's rescaled. All right, this is great. It seems to be a way in which then this could be used as a ruler to know exactly how much you've grown and you need to grow. But the question then became, how do you build a system like this, with molecules? And so this is the idea. I'm a little worried that I'm going a bit slow, but let's see. So after you amputate, it's well known that you generate both in the wound, what is called a wound epidermis here, which is a source of ligand, and in particularly the FGF20 ligand or some FGF ligand. There's also some expressed by the fibroblast. And what will happen to this ligand, we think that as the tissue grow are going to get displaced, they're going to be affected by the tissue growing. But this source is also transient, right? So you get it, it's induced after injury, but then about a week or 10 days, it turns off because injury is gone. But if these ligands are sufficiently long-lived, this dying off source can essentially leave behind a really, really long trace. And because most of it is controlled by vection, which is itself controlled by proliferation, if you scale this thing correctly, you can get something that's perfectly scaled. So this is the idea. Together with my long-term collaborator, Massimo, we put those ideas into a model. So essentially, we're saying ligands are produced at the distal end, the tissue is growing, the process lasts about the same in lateral or medial rays. So if we say that ERK is proportional to some density rho that describes some ligand, what you're going to have on the right-hand side here is your classic, a ligand is made, a ligand is processed, and there is a source. But you also have to consider the fact that this ligand is affected and diluted by the fact that the tissue is growing. We need to know what is this velocity field of that, and that's relatively easy for us to get because these osteoblasts do not move. The only reason why they move is that tissue is growing behind them and that displaces them. So the only reason why if I follow a cell, why this cell is moving is because proliferation has happened behind it and has displaced that cell. That means that velocity is nothing other, at least proportional to the integral of the proliferation, but because proliferation depends on ERK activity, I can approximate this with a power law, and then I get an expression for the velocity that I can close. We know that the ligands are always expressed at the tip. Ashley here did an experiment where she sequenced around the tip and near the amputation site, and you probably can hardly see those little bars here. So the ligands are always expressed near the tip, and that's not a surprise. I mean, this was kind of known. This is just our own way to revisit that. We think diffusion is irrelevant relative to advection as long as the diffusion constant is much smaller than a micron square a second. The Peclet number will tell you that it's dominated by advection. We don't really know what the ligands is, what the diffusion is, but from our scale experiment and others, we think that FGFs are not going to diffuse that far. They interact with heparin sulfates and everything. So then we could essentially take the model without the diffusion, solve it, and we can find condition where we can get a pretty decent fit of those data we had. Yeah.
What do you use for J?
What we really did, honestly, in this simulation is that you can invert this problem.
Because J is a function of location from the tip, since it essentially is localized.
So we could do two things. We put a J that it covers about 15 to 20% of the tip. But you could do another exercise, which is, because we have measured rho, and we have a mathematical expression of these, the only thing that we don't know here is J and tau. So given a tau, I can fit J, and I can get that a transient localized source is sufficient to explain my data. So in a way, you can solve the inverse problem, saying, like, given this data, what is a source that would be compatible with it? And you can get a source that is localized at two conditions, which is essentially this whole model works, but there's two things that need to be true. The first one, which is probably the one that might be more surprising, is that this only works if these ligands are really long-lived. They must, and this doesn't necessarily need to be literally ligand lifetime. It could be persistence of anything, right? Persistence of a cell state, or the ligand could be recycled. All of that would be compatible. It's just a general lifetime. But essentially, signaling needs to persist for about 4 days or so. And the other reason why this model gives you scaling is after you cut, you generate an initial domain that is bigger, signaling domain that is bigger. Then that domain will set up more proliferation, which will stretch the gradient more, and that will stretch it more. And essentially, as long as you have set up this initial length scale or the length scale of that source, if that scales with that lamp, then everything is going to scale, because that's not really the only length. The only length we put in the problem is the length of that source. So then everything scales with that. So this is come up as a very, very simple. You don't need any fine tuning. I think Lisa has a question too, but probably.
After you finish.
Yeah. So essentially, this gives you scaling for free. So you don't need any fine tuning or anything special. But these two things must be true. Yeah.
I'm trying to think about dimensionality of the problem, and it seems like this is like a one-dimensional problem you've solved, but there is another dimension that is into the plane. Like, don't fins get thinner with, like? Okay, so
that's what you are saying is going to be crucial for the second part of my talk. It's absolutely the answer for the mesenchyme, but osteoblasts are essentially one sheet of cells.
Okay, so
and the width doesn't matter. Okay. The height does matter, but it matters for the mesenchyme, which is sandwiched between the bone. But the bone that has no width. It's essentially two cell layers, one above the matrix and one below.
So, like, the fact that you've set up a system which is a one-dimensional track is somehow helping this mechanism work very robustly.
Yes, it will not work, and I'll show you that mesenchyme we think works completely differently. Yeah, cool. So that's a good.
Can I ask another question? Sure. In that reaction diffusion system that you have, there's a characteristic scale, which is the square root of v tau length scale. There is another characteristic scale which comes from v and tau. Yeah. And there's a third scale which comes from balancing j and d. Which one of these controls the speed of the regeneration?
We think it's v tau. It's mainly determined by advection, essentially.
So other two scales are too small?
So we think that d over tau, the diffusion, it's too small. And this is we are dealing with gradients that are millimeters long, and our back of the envelope estimate suggests you either need extremely long lifetime or extremely fast diffusion. It will be very hard for D over tau to be controlling. And J is very transient and very localized at the tip. And so we don't think it can give you these long gradients. But what gives them is the combination of advection and a very long lifetime.
And which one of these parameters is varying when you go from the midline towards the lateral region?
So that the source is changing, but that sets the advection, right?
So you're saying V is changing when you go from
both J and V change at scale, yeah.
Then you're saying that J doesn't control things.
Well, I think J and V, you cannot decouple them, right? Because the growth comes from, so in a way it is J, but it does it through V, right? So because, and this is the point here, if you get a bigger source, you're gonna get more advection, which is gonna display things more. And then the source disappears and you still get a little bit of growth and that drives it out. But that length scale, the fact that it's a millimeter long is not set by the spatial property of J, which is only localized, but V will continue because the ligand have a long lifetime. Yeah.
The source itself, you said it's transient. What's the time scale on which it decays?
I'll show you in a second. So it's about a week, so it's faster. So okay, so is or maybe I might not have the data there, but it disappears about a week before the signaling disappears, at least, if for one of the ligand that I'll show you about. So measuring the lifetime of the ligand or this persistence of signaling has been hard. What we can do is we can block protein synthesis with cyclohexamide. And if you look at a control versus an experiment, ERK activity is still there, right? So clearly there isn't a lifetime of 3-4 hours. You could block protein production for 12 hours. We have verified that we block protein production with photoconvertible proteins. And you go there and there's still as much ERK activity in your control as your experiment. That says there's a lifetime of at least a day or two. It's not too far from 4. We haven't been able to tag the FGF ligands. We have tried Western, we haven't got it to work. So this is the best I have evidence for now. But we have a little bit better evidence on this point at the source scales. And the reason is that at least we know there is one FGF20 called FGF20A absolutely required for regeneration. Without it, you get no blastema at all, no regeneration, no growth. What is interesting is that it's expressed by the wound epidermis, and we have a transcriptional reporter for it. So we can go with our line, image the skin, and image all the cells that express these ligands, and then we can just count how many cells are expressing this FGF ligand. And when you do that, you see that there's a good correlation. So the more tissue you remove, the more FGF20 expressor you remove. Okay, is this just a correlation? Hard to do an experiment where you manipulate it directly, but the genetics is useful here because there are these long fin mutants that make very long fins, as the name will probably say. But when you cut them, they grow a lot more like wild type. They seem to have lost memory, right? This medial ray overgrow, but this lateral ray don't. And if I'm correct that all the memory is the number of FGF20 cells you have, this should have a messed up number of FGF20 cells. And sure enough, when you measure it, you find that for this medial ray or for shortcut, they do fine, but then eventually they just plateau. So the number of FGF20 precursor you get, it seems to reach about 5,000 and plateau. But what's interesting, instead of saying what is the relation between this number and the length amputated, I can say what is the relationship between that number and the tissue that grown 4 weeks later at the end of regeneration. And now things magically correlate again. So this mutant overgrows by about 50% compared to wild type. So it's doing something on top. Our impression is that the lifetime of the ligand now is gone from 4 days to even a little longer. That would be the simplest way to explain this. But sure enough, now for all this red point, I can kind of predict how much tissue that will regenerate by how many FGF20A expressor have. So this really leads to this idea that there is some memory set up very, very early on, and then there's a program that just scales that initial source. All right, so this is where we got for the osteoblast. About fibroblast, this is the work of a great student, Alvin. And what Alvin at first said was like, okay, let's see if the fibroblasts follow the same rules. And honestly, at first, I thought, okay, probably we're gonna find that everything's about the same, right? Same pathways. The geometry here is a little different. So it's essentially all this mesenchyme that fills in between the two bones. So you can kind of see this nice layer here. That's the bone. This is all the tissue in between. This is an XZ section. There is a tip that is non-proliferative and non-proliferative niche, which expresses or responds to Wnt signaling. This is a Wnt signaling reporter. And then there is proliferation that follows behind. So Wnt signaling is important. And so again, is FGF-ERG. So we decide to look at both. Should probably introduce this. So what Alvin does now, he does two cuts. He does or different cuts. Instead of cutting at the same height, he cuts perpendicular because what he wants to get some medial rays in which he has removed about as much tissue as a lateral ray so that he can then compare which property are dependent on the identity of being medial and lateral and which one honestly depend on length amputated. And what he discovered is that pretty much Wnt signaling seems to segregate on which ray you are. So the lateral ray activity seems to segregate differently than the medial ray activity. And there was really little evidence that the total amount of Wnt signaling depended on length amputated, but it seemed to somehow depend on geometry, and we'll get back to it in a second. Things were slightly more interesting maybe for ERK and proliferation because what he found there is that there is in fact a correlation. So you have more proliferating cells and you have more ERK positive cell and more proliferating cells as a function of length amputated. However, what confuses us for a bit is that the size of both the ERK domain and the proliferation domain was fixed. It was always about 500 micron. So if I cut 5 millimeter, I get 500 micron of proliferation. If I cut 1 millimeter, I get 500 micron of proliferation. Yet I get a lot more cell in one case than the other. The average seems about the same. That doesn't make sense unless, as Lisa was saying, you remember that it's a three-dimensional tissue, right? So okay, what about the width and the height then? So when Alvin looked, and he can make these really nice data sets where he can look at osteoblast, fibroblast, and get all the geometric property. So this is going to be the width of the fin, and then if you turn it around, you'll be able to measure height. And when you look at this, the width is just a property if you are a lateral or a medial ray, it's kind of fixed and segregates. But instead, what segregated very, very nicely with the height was what segregated very well with the length amputated was the height. So that now a lateral and a medial ray that you have cut similarly are going to fall on top of each other. But if you cut more, you're going to have a taller blastema. And if you cut less, you're going to have a shorter blastema. That is so true that if you actually measure the height at the tip, let's say 5 days after regeneration, and you plot it versus length amputated, you get this really, really nice relationship. And what really was very exciting, probably the most both surprising and exciting thing that Alvin discovered was that he could actually do even more. At every time point, he could essentially take the height of the blastema and predict the growth rate at which the blastema is going to elongate. And he can collapse all his data on this one unique relationship. So if you tell him how tall the blastema is, he's going to tell you how much it's going to elongate. That maybe seemed too good to be true, so we went in and did this treatment with a drug that inhibits calcineurin, and those that tend to overgrow. And there's a bunch of measures here that you can ignore. This is the main plot. He again can take all this data from the drug and put them essentially in a single relationship with all the wild-type data. And again, if you tell him how tall the blastema is, he can tell you how fast it grows. Of course, if... Olivier, do you have a question?
Yeah. I'm not sure I understand how do you define the height of the blastema?
It'll become clear in a second, but what you do see when you look at these, sorry, it's the next one. Yeah, it's a little tricky because it's a position dependent up to a point. So you see that it increases, but then it stops and plateaus. I'll skip ahead, sorry. So what you see is that essentially it grows in this kind of triangular shape and then it stops growing and it's more or less flat. So we define that operationally. as the height. Yeah. Okay, so these work quite nicely, but of course the rate of growth slows down, right? And you can also see here that the height decreases. So Alvin wanted to figure out what is the law with which this height decreases over time. Yeah.
And the ray is also thinning.
Yeah, we'll get there. So I think that's where the information is essentially. Yes, that it's at the end of the day. Yeah, they're reading what was there to start with. So and this is also interesting is that the rate at which the tissue is tapering or reducing its height also seems to correlate more or less with the height. So when I put these three things together, essentially, it seems that these are, there's 3 Ohm's law for how the fin grows, 3 linear simple relationship. The growth rate is nothing other than the height minus an offset. There is an exponential decrease of the height over time. And there is if you look at the initial blastema that is set, that height scales exactly with length amputated. Once you put these 3 things together and you solve them, you can explain why different rays grow different amount and the rate at which they grow pretty well. So now this, of course, poses the question of how do you generate a system that follows these 3 rules, right? So the first one is how can the growth rate be controlled by the height of a tissue? Here you can write the simplest model. The increase in volume is nothing other than proliferation, and they are only extending in 1D. Right? Let's say that the width and the height don't change because they change very slowly. Then if you put all of this together and you say that this is the integral of proliferation and proliferation is fixed in its width and length, then what you get very easily is that that the growth rate is nothing other than the average proliferation in Z. And one way in which you can get that the LDT depend on H is essentially if there is a gradient of proliferation in Z. If there is a gradient of proliferation in Z, you'll get that dependency. And Alvin went and looked, and sure enough, there is such a gradient. What you're looking at here is what is the probability of proliferating as you move away from the boundary, and there is this very, very clear gradient. Essentially, what is going on is this. The proliferation is inhibited at the boundary, and it grows as you move away from the boundary, but the taller you are, the higher the probability is going to get. And so the taller you are, the more you're going to proliferate, right? So essentially, they're going to follow the same curve, but for a small height, you're going to reach the middle and you're going to go back down. For a large height, you're going to grow larger. And even once you rescale them, then the large height is going to have more proliferation than the little one. This is just all you need. If you have this gradient, you are going to get the proliferation depend on height. That gradient does not seem to correlate with a gradient of work activity. So we think there's another pathway. We don't know what that pathway is yet. We really hope to find it because I think it will be a new contribution. We have some ideas and speculation of what we will be. So hopefully next time you hear me talk, you'll hear something about it. The other thing that is interesting is what controls the height, right? So the height has to change appropriately over time to control the growth rate at the right time. And what I was already finding out is that it looks like this height is growing, and then at one point it stops growing. And you can clearly see that if you look at the height in a given position, it's fixed, and then it's almost like something is coming and dragging along. We don't know yet, but one interesting observation that Alvin had is that the point where they stop to grow seems to be exactly where the bone ends. So the bone is growing on top of the fibroblast, and essentially where the bone ends, that's where the tissue stops growing. But for the tissue to decrease in height, that also means that the bone has to grow a little faster than the fibroblast so that over time it kind of closes in. We don't know what that is, but we have never seen any loss of bone cells, but we see that there is a little bit of loss, probably, we don't know if by apoptosis or cell shedding at the tip of the fibroblast. So it's possible that cell death could contribute to this thinning. And then the last thing in the last couple of minutes is what sets this initial height. And of course, Olivier already hinted at the crucial point, which is what is even the height of a fin before you cut it. And there's 2 things that are interesting here. One is that this relationship is different from the other one, so it's a bit more quadratic. However, we can collapse pretty much all our data on a single relationship if we plot the height as a function of the distance of the tip. So this system has built a grid, a system of coordinates that tells you if you are at such a distance from the tip, your height is fixed. So it doesn't matter if you're a medial or a lateral ray. If you are 2 millimeters away from the tip, you're going to have this specific height. And that seems to be quite reproducible. But then what needs to happen is that the system needs to be able to take this height and transform into that height that will sit on a different curve. So how would you do that? We do not really know what the signals are, but what we could do is to look at our proliferation reporter and look at how proliferation changes. This is very early on. This is proliferation only at 2 days, so sorry, this should be at the 2 days post-amputation. And you can look when you cut 700 micron to all the way to 6 millimeters, how this proliferation is different. And we can quantify, but what is very interesting is that we can take this two relationship I've shown you before, and then I can say, if I really understand what is going on, because the maximum of proliferation is the same, the only thing that should be different is the proliferation domain. But since I know all these geometric parameters, I should be able to predict exactly how much proliferation is there by taking the ratio of these two geometric quantity. And when you do that, you get something like this. So the proliferation domain we measure at the point, and the black curves is the one that we just predict from geometry. So it seems to go pretty well. Now, what sets this proliferation domain, we don't know. Early on, since it's so early after an amputation, the natural candidate are things like reactive oxygen species, calcium, something that's really activated acutely following an injury. We don't know that yet. That's pretty much where we got. So I guess for this fibroblast part, I think I could leave you with these three simple linear rules that explain, and what is left us to do is to figure out what are these molecular pathways, what controls these geometric changes, and what sets up this initial domain of proliferation. Right, I can thank everyone. I think the only one I didn't show a picture of is Ashley, who she led all the projects on the osteoblast growth in fin regeneration. And actually, the more recent work on the geometry of the blastema is also with Boris, and Ken is my long-term collaborator with whom I've done most of my fish regeneration work and funding sources. Thank you.